We have osimertinib, and it's a third generation EGFR TKI, and it's a standard of care these days, and it has certainly also a good CNS efficacy, but it's a CNS efficacy sufficient with osimertinib. But the fact is, in development is the fourth generation EGFR TKIs, which are designed to be better CNS penetrant than the third generation. Lung cancer is a kind of a role model for personalized medicine and molecular testing before decision of treatment. And we have many targets. We have many generations of targeted therapies, and I will touch upon a few of them here. But let me start with molecular targeted therapies and early stage disease. And there are two molecular targets which led the way. First, we heard about EGFR positive patients and the Audora study, which was adjuvant osimertinib versus placebo in early stage disease. And as we heard from Tony Malk, there were clearly superiority in disease-free survival, and there is also an superiority so far in the overall survival. And the ALK was the next target, and we had the ALINA study in ALK-positive patients, and we can see adjuvant alectinib did significantly better and is now a standard of care. We have osimertinib, and it's a third generation EGFR TKI, and it's a standard of care these days, and it has certainly also a good CNS efficacy, but the question is CNS efficacy sufficient with osimertinib? As a standard of care now, we have osimertinib and chemotherapy according to the FLORA2 study. But the fact is, in development is the fourth generation EGFR TKIs, which are designed to be better CNS penetrant than the third generation. And there are preliminary data also presented at ASCO this year showing actually that fourth generation EGFR TKIs are very good in the CNS system. Then we have stage 3 disease, non-resectable disease, which I talked about previously was resectable, now we're non-resectable, and we have the LAURA study with EGFR, as we can see here, the LAURA study in resectable disease was clearly in favor of osimertinib. We have other targets and drugs in development for stage 3 disease. At ASCO last week, we learned about the libretto study for RET-positive patients with selpercatinib, and that was also clearly better than placebo in early stage disease. So what I'm trying to say here is we are moving very quickly towards more and more targets and more drugs in early stage disease. So several questions comes up, of course, with this development. We are dealing with some very rare mutations, 1 to 2% of the population, Is a randomized trial needed for every actionable mutation? That needs to be discussed. And what the degree of activity, response rate, duration of response is needed in advanced disease to move it into early stage disease. We do have event-free survival as an endpoint for those studies. But what patients, of course, are most interested is, do I get cured with this treatment? So still we are waiting for survival data, and but as we saw for EGFR, we're starting to have very long-term observation. What is the optimal duration of adjuvant treatment? Patients in ADORA study, they are actually treated until progressive disease, or over several years. We don't know on this stage what is the appropriate treatment length in an adjuvant setting, and hopefully with liquid biopsy, we will be able to figure out how long treatment we need, whether we should escalate or de-escalate over time. What is an appropriate comparison arm is still a question. Should chemotherapy be required or not in an adjuvant setting? That is things we are questioning, and we need answers to in the future. Immunotherapy, as we have heard, it was a breakthrough in many tumors, including lung cancer, and we are still dealing with one main problem, in my opinion, and that is only 40% of lung cancer patients will have significant benefit from IO. And how do we select the right patients? That is something we are working on, but we don't have a very good answer to it, to be honest. We know that PD-L1 immunohistochemistry is useful and are used in clinical trials, and it's also used in the FDA approval of drugs and drug combinations, as we can see here. We are using cutoff of 1%, cutoff of 50%, and we can see here some data with immunotherapy. Monotherapy, this is advanced disease, I have to say, but we can see here that for patients with high PD-L1 expression, monotherapy is good, and we don't gain much with adding chemotherapy in general. There are several studies having shown that in patients with PD-L1 expression below 50%, it is clearly that combination therapy is better than monotherapy. But again, when we are talking patients with high PD-L1 expression, chemotherapy in general doesn't give added value, but more toxicity. However, I should say there are subgroup of patients which we would add chemotherapy. If you have a young patient with a high tumor burden, we will add chemotherapy in many cases. But here is a paradigm we are using these days. PD-L1 more than 50%, single treatment with IO, less than 50% chemo plus IO, and for patients less than 1%, we will either give chemo and IO or chemo plus double IO and PD-L1 CTLA-4 inhibitor. No, we need to move immunotherapy to early stage, and Tony Mock gave a very good overview of several treatments here, and I don't think I will go into all the details as a repetition, but we have still more work to do, particularly also in unresectable stage 3 patients where Pacific study was leading to consolidation durvalumab after concomitant chemo-rad. And that is our standard of care today, and that is what we are building upon. We know that PD-L1 level tells us something about prediction of immunotherapy, as I mentioned, for advanced disease. It is the same for early stage disease. If you have high PD-L1 expression, you seems to do better. The question is, of course, with lower PD-L1 expression, and particularly with negative PD-L1 expression, what are we doing there? And there we seem to have a good results with combination of the IO treatment, as I said, PD-L1, PD-1, in combination with CTLA-4. But we have also in early stage disease new combinations in the NeoCOAST study, which Tony also mentioned. We are including new drugs in combination with immunotherapy, and we have also EGFR moved into the neoadjuvant setting in the NeoAdora. And as Tony mentioned, the data for neoadjuvant EGFR is encouraging, but not, in my opinion, as expected. We would see better complete pathologic response. Now, across neoadjuvant, perioperative, and adjuvant therapy, it is clearly benefit of immunotherapy for resectable or resected lung cancer, also in the cases where we have PD-L1 negative tumors. So we are still questioning the PD-L1 negative space, which is around 20% of the patients, but we will offer early stage adjuvant, neoadjuvant, adjuvant, potentially treatment also for those patients. But we have questions to solve. Should adjuvant immunotherapy be recommended to patients with PD-L1 negative? We need more studies. Should neoadjuvant therapy be offered to PD-L1 negative patients? And what is the role of perioperative, where we give both neoadjuvant and adjuvant treatment? One question we are left with, in my opinion, and which is very important, is what are we doing after neoadjuvant therapy? Neoadjuvant therapy is a standard of care, in my opinion, very clearly in lung cancer. Some patients, about 20% in US, a little bit higher actually in Asia, of reasons I don't really understand, but about 20% will have complete pathologic response. Do they need more adjuvant therapy? And what about those with non PCR. What kind of therapy are we offering those patients in the future? And there are trials trying to address this. This is an ETOP trial for patients with non-PCR, and this is a trial I'm personally much involved in, SWOG-initiated trial, the INSIDE trial, that is for patients with a complete pathologic response. They are given either adjuvant IO therapy or surveillance. So those are the type of questions we are dealing with, and we need studies for giving us the answers. Neoadjuvant or perioperative immunotherapy is associated with increased benefit compared to adjuvant only for resectable and is a preferred treatment. And again, we need to have a better understanding of what will be the post-surgical approach in the different situations. And hopefully here, liquid biopsy can be helpful in the future, which will also be discussed in this meeting. So that is my part of this question. And the next speaker to talk is Dr. Oscar Arrieta from Mexico. Oscar is director for the National Cancer Institute in Mexico. And Oscar, what is your perspective?
First, thank you very much for the invitation. And your first question about the oncotarget mutation, actionable mutation, if we need clinical trials or in order to define if we need a clinical trial for adjuvant treatment, I think that not all actionable mutations have the same magnitude in the benefit. Some will not have an effect on with survival because in the recurrence could lead to we can give the treatment during the recurrence. So I think that is very important to development clinical trials in adjuvant for every actionable mutation.